AIDS. Immediately, scientists began to fight what they soon realized was the most formidable microbe in history.
This virus is not like anything we've seen before.
It's an incredibly difficult problem. People in laboratories all over the world, brilliant minds, have been addressing this problem for almost a decade.
After $10 billion and 13 years of research, scientists are now confronted with the surprising convergence of two lines of investigation. The fact that some people fight back the virus, and that a vaccine made from the live AIDS virus looks promising. But is it too risky to test?
It's crazy to think that we're going to solve this problem without taking risks along the way, and frankly, that we'll ever be able to solve it without losing some lives in the battle.
Tonight, a special report on that battle, the losses, the victories, and the future. AIDS research, the story so far. AIDS research, the story so far, is made possible by the Aaron Diamond Foundation, supporting projects in New York City in minority education, culture, and medicine, including AIDS research, education, prevention, and the civil rights of people with AIDS. These test tubes contain one of the most deadly viruses known to mankind. Should scientist David Ho accidentally inject himself, he would probably die. Yet the virus he is handling is in the bodies of 1 million Americans and 14 million people worldwide. Most of these people will go on to develop AIDS and die. Nearly $10 billion have been spent on scientific research fighting the war on AIDS. This program is a report from the front lines. AIDS broke out 13 years ago among the gay community and spread to all parts of society.
I have known since the time of my brain operation in September 1988 that I have AIDS.
Because of the HIV virus that I have, I will have to retire from the Lakers.
AIDS wasn't confined to America. Its biggest impact has been on the developing world.
In Central Africa, there's a term that's applied to AIDS, which is called Bahara. I think the scenario is that in the next millennium, after the year 2000, tens of millions of people will die from this.
I know that there are drugs out there that can save my life, and I want to know why they're not being tested more quickly.
What we're starting to see now is a pattern in Asia, particularly in Thailand and in India, which tells us that the next major catastrophic wave of the epidemic will be in those Asian countries.
Many times in the past 13 years, dying AIDS patients have had their hopes raised, only to see them dashed.
Is it a breakthrough or ballyhoo?
I would encourage AIDS patients to see anyone who is able to give cyclosporine, which is anyone in the world.
He's been cured?
Because the last test made in America shows he has no AIDS anymore. They call the drug GLQ223, or Compound Q.
I'm excited about our experimental findings, and I can't wait to see it in clinical trials.
Blood is taken from the patient, heated to 108 degrees Fahrenheit, and returned. The theory is that the heat destroys the virus, much as a fever combats more common illnesses.
Today, researchers at Massachusetts General Hospital in Boston announced the single most promising scientific AIDS breakthrough in 7 years.
Only time will tell whether this involves a turning point in HIV.
But in 13 years, there were no turning points, and there were no cures. Despite great advances, the scientific community has found HIV, the virus that causes AIDS, a much tougher opponent than they had originally thought.
in 1984-85, many people thought, perhaps naively, that we should have a vaccine in a matter of a few years, and we should have a number of drugs also very quickly. Certainly now in 1993, looking back, that was very naive. This virus is not like anything we've seen before.
Despite their huge resources, even the world's pharmaceutical companies have so far failed to conquer AIDS.
It's an incredibly difficult problem. People in laboratories all over the world, brilliant minds, dedicated workers, have been addressing this problem for almost a decade. And it's just an incredibly difficult problem.
Scientists now know more about the human immunodeficiency virus than any other virus. It enters the body in blood or seminal and vaginal fluid and seeks out a key cell in the immune system, a white blood cell called the T helper lymphocyte, 150 times its size.
They have on their surface a specific doorway, a specific structure through which this virus can attach and enter. Like many unwanted guests, once it comes in your door, it decides to take its coat off and make itself at home, and that's what it does. So it enters the cell at the surface, it sheds its coat, and then it sheds its inner core, revealing its genetic material. In order for the virus to insinuate its genes within your genes, they have to be in a DNA form, not an RNA form. So the virus makes a DNA copy from its RNA base or template.
This DNA then could move into the nucleus of the cell and then become part of the cell's chromosome. So the virus, in essence, integrates itself into the cellular chromosome and becomes part of the cell.
The viral genes now take over the machinery of the infected T cell to reproduce itself. And then the virus migrates to the surface of the cell and buds out from the cell membrane. Is now a free new viral particle which can go infect an uninfected cell and start this whole life cycle over.
Before it is destroyed, one infected cell can produce thousands of HIV particles, which go on to infect other lymphocytes. Despite huge amounts of virus, all most infected patients feel is a bad case of the flu.
During the flu-like illness, there's enormous amount of virus in the bloodstream and probably in lymph node and other sites in the body. And in fact, the level of virus during this period is often higher than what you would find in a patient with terminal AIDS. But the immune system kicks in very quickly at that point and brings the virus under control in the course of just a few weeks. And it's very, very dramatic.
There's now a period where you feel fine. And that can last from a year or two, and then there are some people where it seems to last many, many years, a decade or more. Over this time period, though, the virus begins to chip away at the immune system. As the immune function falls below a level of 80% loss or more, people become predisposed to major infectious diseases.
No problems with
Pneumocystis pneumonia, tuberculosis-type organisms, toxoplasmosis, which causes an encephalitis, and so on. And they become predisposed to certain cancers, lymphoma and Kaposi sarcoma. Now, this was clinically called AIDS. Deep breath. Great. Looks good. Sounds great. Lie back now.
Physicians like Groupman treat their patients with antibiotics to control infections and with drugs designed to interrupt the HIV life cycle. The most famous of these drugs is AZT. When news of AZT broke 8 years ago, patients' and doctors' hopes were raised.
John is taking a new drug called AZT. This synthetic chemical doesn't cure AIDS, but it can stop the disease from getting worse. AZT confuses the AIDS virus, causing it to stop multiplying, which means it stops destroying the AIDS victim's immune system.
I take walks, I dance, I'm alive again.
After entering the cell, the virus must convert its RNA to DNA. AZT targets the enzyme which enables this transformation. Without this critical step, it cannot get its genes into the cell's nucleus. Unable to turn its genetic information into a DNA form, the virus is effectively disabled. But within months, mutant strains of HIV emerged that were resistant to AZT. These strains were able to complete the life cycle. The virus continued replicating. The patients eventually got AIDS and died. Their lives extended a year or two at most.
The problem with AZT and with all of these drugs that work at this point in the virus life cycle is that the virus is very clever and that it mutates and is able to become resistant to AZT. And generally the benefit is measured in a matter or in a time scale of months to years.
Scientists now realize that HIV's phenomenal ability to mutate gives it the power to outsmart most drugs.
The virus goes into our bodies, replicates prolifically, and seeds itself throughout the body before the immune system can catch it. At that point, a lot of variants are already planted all over the body, and those are the ones that eventually show up when you treat with drugs. They're already there. They're not that they appear, they're already there, and in some cases, that's why the infection springs back, especially with some of the later drugs, within weeks of first treatment.
Frustrated and disappointed with AZT, scientists are turning their attention to other parts of the life cycle in the hope that they will find the virus's weak spot. Many of the major pharmaceutical companies are very enthusiastic about a new class of anti-AIDS drugs called protease inhibitors. These drugs, which they hope will revolutionize treatment, target a critical enzyme in the HIV life cycle. The enzyme, called protease, acts like chemical scissors to cut a long protein into pieces, which can assemble into a new viral core. If one could block the scissors, the virus would be unable to infect other cells. To find out everything they could about protease, scientists at Merck crystallized it and X-rayed it from many different angles. This generated a picture of the position of all the electrons in the crystal. From this pattern, they were able to generate a three-dimensional computer model of the protease molecule. Knowing the shape of the protease, the team then designed a drug, a protease inhibitor, that would fill the hole. With the chemical scissors blocked, HIV is unable to replicate. In early studies in the test tube and in people, the drug worked very powerfully. But the scientists are not overconfident. They are fully expecting the emergence of mutant forms of HIV that will escape their drug.
Resistance is the issue now for this disease. It's clear that a number of laboratories, including our own, have developed very potent antiviral agents that will completely stop the replication of susceptible virus. But this virus is incredibly adaptable, and resistance is the issue for coming up with a long-term, clinically useful therapy.
And this time, the scientists intend to fight back by designing a new protease inhibitor to block any mutant proteases that arise. And should yet other mutant strains emerge, they will design other protease inhibitors to block them. In this way, they hope to keep one jump ahead of the virus. Even the most enthusiastic researchers admit that protease inhibitors will not cure AIDS. But in conjunction with AZT and antibiotics, they might extend a patient's lifespan considerably.
if we really succeed with the protease inhibitors, one can imagine being able to tell someone with HIV disease that they could live for decades, that we can change this from a fatal illness with progressive immune deterioration to a more chronic and controlled condition, and hopefully tell a 25-year-old that they can see their 50th birthday or beyond, which I think would be an extraordinary contribution.
But some scientists have learned not to be too optimistic.
In studies in vitro and in early studies in humans, the drug looks reasonable, promising as a matter of fact. But I think it would be foolish to say that this is the great new promise of this drug until we see a lot more people being treated with it.
Should protease inhibitors prove effective, they will have to be taken several times a day for life, and they will not be cheap. Along with AZT and antibiotics, a single AIDS patient's drug bill might exceed $10,000 a year, a figure completely out of reach for the developing world.
In the developing countries, we're essentially talking a clinical language which is not understood. The per capita yearly expenditure is on the order of $5 or $10 in Zaire. We had a visiting group of African clinicians from Central Africa come here and spoke with them about new advances in treatment and so on. And they said, we don't have aspirin, we don't have bandages, we don't have AZT is too expensive for us to distribute on a widespread basis. So I think that realistically and sadly, patients in the West and in the developed world will have access to these kinds of treatments. And patients who are in the developing countries will not.
And tragically, the developing world is where AIDS is spreading fastest. The latest data shows that in some countries, AIDS threatens to consume a large part of the population.
We have a situation in sub-Saharan Africa right now where the epidemic is truly out of control. The prostitute populations in some of these countries, 50, 60, 70% of the women are infected. A startling figure is emerging out of the northern provinces of Thailand around Chiang Mai, where everyone who's a male and 21 years old gets drafted into the service, into the army. And what we've seen over the past couple of years, that just a year or two ago, the number of otherwise healthy 21-year-old recruits in the northern province, over 10% of them were infected when they went for their draft physical. Now, in certain provinces and northern areas, that is approaching 15 or more percent, and some even as high as 18%. That's astounding.
In many cities in Africa, over half of the entire population is infected with this virus, and probably the vast majority of those people will die in the next 5 to 10 years. That's a devastating situation that exists today and is only getting worse in many parts of the world. Certainly we're seeing tremendous explosion in South America and in Southeast Asia and in India, and we're likely to see that continue. And I think we're about to see an explosion in Eastern Europe and the Soviet bloc countries as practices change and they open to the West.
People in the developing world can't afford expensive drug treatments for HIV. What they need is a cheap vaccine to prevent infection in the first place.
The ultimate ideal that we would all love to see attained would be a safe, highly effective, cheap vaccine that you could give one shot and you were fully protected for life. That was cheap enough that could be used worldwide. That would be the ideal that we strive for.
Vaccines are one of the great success stories of 20th century medicine.
Historically, vaccines have been our most potent weapon against viral diseases of medical importance. It includes poliovirus, which was the scourge in the United States in the early 50s and late 40s that all parents were afraid their children would get. Smallpox was never that serious a problem in the United States, but worldwide was a tremendous problem. Due to massive efforts of the World Health Organization in the 1970s, smallpox has been eradicated from the face of the earth. It's the only viral disease that's ever been totally eradicated from the face of the earth.
But finding a vaccine for AIDS has not been so easy, because HIV is not like other viruses.
The problem is, how do vaccines fundamentally work? Most people think vaccines prevent infection in the first place, but they don't. What a vaccine does is it prevents the infection from spreading to cause the disease. It relies on a rapid recall of the immunity to shut down the infection at an early stage before disease occurs. In other words, we let the invading organisms in, but wipe them out early. That won't work with AIDS. Once the infection is in, the damage is done.
The virus, unlike virtually any other virus that we deal with, has the capability of inserting itself into the genome of a cell and hiding in a latent form, so that a substantial amount of the viral burden is in a form that isn't even recognized by the immune system. So it doesn't even have to mutate, because the immune system can't see it anyway. It's hiding there.
For this reason, an HIV vaccine needs to act quickly and neutralize free virus with antibodies before they enter the cell. But there is another problem. HIV is not a simple virus like smallpox. It comes in many different strains. An effective vaccine must be able to protect against all of them.
With HIV, all these different strains are running around at the same time. You're not talking about one strain this year and another strain next year. You're talking about they're all present at the same time. Even in one infected individual, there's a complex mix of thousands, tens of thousands, certainly tens of thousands, probably hundreds of thousands.
And HIV strains in patients in San Francisco may differ by up to 30% from the strains in African patients. Despite these formidable challenges, over 20 vaccine projects are in progress. They are coordinated at Duke University under the direction of Danny Bolognesi.
Let's take the easiest possible case. Let's go into an area where the viruses are homogeneous as possible. Not this 35% difference, obviously, much less than that. Let's use a single virus that best reflects the properties as we know them of the viruses where we're going to test. If we get some efficacy, if we're able to show something, then we can modify our approach into a cocktail form and add this and this and this and try to cover the waterfront.
Out of the thousands of strains, certain field strains were selected and grown up in the laboratory in culture for a time. This yielded very homogeneous strains of virus. Scientists took parts of such viruses, such as key molecules on the surface which trigger immune responses, and made vaccines, which they injected into healthy, uninfected volunteers. Being made from only part of the virus, they were safe. The question was, did they offer protection? Sera from the volunteers is sent to Duke University, where it is stored in this freezer, awaiting testing. What they found was extraordinary. When Sera was challenged with the same lab strain of HIV from which the vaccine was made, antibodies in the sera neutralized the virus and killed it. But when they challenged it with wild strains of HIV found in patients, similar to those from which the lab strains were derived, the antibody had no effect. If a vaccine failed to protect against related field strains, What chance is there that it would protect against the thousands of other wild strains? Scientists were shocked.
It was a surprise, quite frankly. It was, I think many people anticipated that neutralizing viruses in that setting might not be as easy as doing it here, but a complete negative was unexpected. Was a surprise.
What I can't tell from this...
The negative results have thrown the entire U.S. vaccine effort into disarray.
Think of it this way. If your strategy, if a vaccine strategy is based on neutralizing antibodies against the virus, and the news is bad that what you're inducing by a particular vaccine really does not neutralize the viruses, and you're sure of that, and that you're unable to do that, what's the rationale for that vaccine?
What's the point in going ahead?
Correct.
Charles Schrader, in the action. It's time to put up or shut up, Bill. Put up or shut up. A Manhattan Project on AIDS. Research needed now.
People are frustrated. After 13 years and $10 billion spent on research, there is no cure, and a vaccine seems unlikely. But there is one possibility. Ron DeRosier has an idea that might work if the scientific community can find a way to try it. DeRosier first became involved in AIDS research by accident. In 1980, some of his monkeys started dying.
They were dying with an assortment of infections that they shouldn't have, that shouldn't be killing them. Things like generalized CMV, and they also had this severe weight loss and wasting syndrome. It was a mystery to all of us. We didn't know what it was or what could be causing it. And it was only then, in 1981, that the New England Journal article appeared describing this immunodeficiency syndrome in people.
DeRosier realized that here was an animal version of AIDS. Monkeys contracted simian immunodeficiency virus, or SIV, that was strikingly similar to HIV. In 1986, he set out to develop a vaccine. He tried dozens of different kinds of vaccines made from subunits of the SIV virus, using all the strategies currently being tried with human vaccine trials. But as with the tests at Duke, nothing worked.
We faced dismal failure after dismal failure. We've had very poor success in being able to achieve protection in the laboratory. They just didn't work. And what was most disappointing about those results is that we did everything we could think of to stack the deck in our favor, as unrealistically as you can imagine, to try to get a success. And even when we stacked the deck in our favor, we couldn't get a success. And if we couldn't get it under those conditions, how in God's earth is it going to ever work out in the field? So that's when I went through my period of a real pessimism that there would ever be a vaccine. It was right around that time then I started thinking we really needed some more radical strategies and maybe even start thinking of some unthinkable things.
DeRosier's unthinkable idea was to make a vaccine from a whole live virus after first weakening it. He noticed that among the virus's genes, there were a few that did not seem essential. By removing one of these genes, the NEF gene, DeRosier reasoned he could attenuate the virus without killing it.
We took out one such non-essential gene, a gene that's called NEF, and to our enjoyment and happiness, we found that the properties of this virus missing the NEF gene were pretty much exactly what you would look for in a live attenuated vaccine.
The attenuated virus enters the cell and begins its life cycle. It replicates, it varies, but it does everything much more slowly.
It seemed not to cause any disease in monkeys. The initial 6 monkeys that were infected with this strain 4.5 years ago are all still alive. They're all still healthy. They have no CD4 declines.
And when de Rosier challenged the vaccinated monkeys with lethal strains of SIV, for the first time, he had success.
Nothing that we've tried previous to that had come anywhere close to the level of protection. Here we were getting 1 dose of vaccine, 1 shot of the vaccine. We come in 2.25 years later, give a large dose of challenge virus, and the animals are completely protected. In 2 of those animals, we actually gave a very large, whopping dose, 1,000 times more virus than we would need to infect an animal. And these animals have completely resisted that challenge. And it's now been more than 2 years.
Despite de Rosier's success in producing a live attenuated vaccine for monkeys, many scientists are nervous about trying out the approach in humans with HIV.
Well, because it's a live HIV, in essence, even though investigators like Ron DeRocher have made specific mutations in the virus to weaken it considerably, there is always the fear that even weakened HIV would ultimately cause immunodeficiency and disease some years in the future. What if the complications occur 20 years from now or 25 years from now? How do we do a proper safety study without a prolonged observation period? And yet we need a vaccine urgently. We cannot be doing safety studies for 20-some years before launching into a larger trial of these vaccines.
It's a tough call. Given how much we want to have an effective vaccine, one has to ask very hard questions about a live virus which is weakened, attenuated. First is, could it in some way revert to become less weak but actually quite potent? Are we really clever enough to make the necessary deletions to so attenuate it that it couldn't revert? Moreover, the viruses integrate into your own DNA in a random fashion. It potentially causes mutations within your own DNA, within your own genetic blueprint, which over time is in a way that cancers emerge. So could this be a mutagenic intervention which ultimately will cause the early appearance of certain cancers? So I think there are a number of serious unanswered issues related to safety.
But in the past few months, some scientists have seen a possible way forward. Their hope comes from a group of individuals who, against all odds, seem to be able to resist HIV. Sal is a resistor. Over 11 years ago, he was infected with HIV through sharing needles. When he found out, he thought his life was over.
I felt very cheated. And there were a lot of dreams that I had had and things that I wanted to do. And one of them was to go back and finish my degree. At the time, I only had about 20 credits, 20 college credits. And going in the evening, working full-time, I was looking at 4.5, 5 years to complete my bachelor's degree. But I refused to allow the virus to cheat from me or to take from me something that I really wanted to do. And so what I did, I think, was channel the anger against the virus and used it as a motivating factor to go back to school and to continue my studies and to continue my work. Sort of was trying to get 75 years of life into what I expected to be a 5 to 10 year lifespan, and just sort of went on automatic pilot. And I subsequently completed the bachelor's, and I even went on and completed the master's, which I'm very proud of.
As Sal studied, he realized he was feeling fine, whereas his HIV-positive friends were dying.
Most of them had an initial period of being healthy and feeling okay, but every time they would go and get their T cells done, there would be a drop. They would go in, it would be 500, the next time it would be 450, and over a period of years they would experience this decline. And I started to realize a couple of years ago that that really was not happening to me.
Sal has never felt better. He has not shown any symptoms, and his immune system is perfectly normal. So far, he is holding the virus in check.
I haven't smoked a cigarette in over 8 years. I exercise. I go to the gym 2 to 3 times a week. I run. I don't expect the bottom to drop out. I feel that my prospects are very good.
Cases like Sal's may transform the prospects for an AIDS vaccine. Several hundred such long-term non-progressors have been identified who can apparently keep the AIDS virus in check, including a very important group that has recently come to light in Sydney, Australia. In 1987, an administrator at the Sydney Red Cross Blood Bank, Jenny Learmont, got a call about a 75-year-old man who had discovered he was HIV positive. He had received a blood transfusion 4 years earlier.
Going back to America? Okay.
Today, all the blood taken in by the Red Cross is tested for HIV. But in 1983, when the man received his transfusion, no such test existed. So Jenny Learmont checked through the records to find out whose blood might have infected him, and discovered that he had received 1 unit of blood from a young man who was later diagnosed HIV positive. Over the next few years, several other recipients of this donor's blood turned up. All were HIV positive. All expected to develop AIDS and die. But as time went by, she realized an extraordinary thing about this group of 6 patients. None of them seemed to be developing clinical AIDS. In fact, their health was fine. Dennis, a 65-year-old man, was transfused in 1984 during gastrointestinal surgery. Like the other members of the group, he didn't want his face shown because he fears the stigma of friends knowing he is HIV positive. Despite the fact that he has been infected with HIV for 10 years, his immune system is in perfect working order. The same is true for the other recipients of the infected blood, like Marion, a 40-year-old woman transfused in 1984. Jenny Learmont realized she had stumbled on a group of people of immense importance to AIDS research.
There's 3 men and 3 women. They're very different ages. They range now from nearly 80 down to 40. They had operations, their operations were all different, so they were transfused for different reasons. So the only common factor about that group is that they received blood from one person. The group themselves are all really well. Their whole immune picture is normal. And they've been infected now the longest 13 years and the shortest 10.
Jenny and her colleagues believe that the donor, who is also in perfect health, must have contracted a weakened strain of HIV, and this is being checked in the lab. If this turns out to be true, the group has unwittingly participated in the first live attenuated HIV vaccine experiment.
If it indeed turns out that some of these non-progressors are infected with a rare occurrence of a mutated strain of virus that's, for example, deleted in NEF or deleted in some other non-essential auxiliary gene, and that there is good epidemiologic data to indicate that these individuals are non-progressors over a long term, then that, I think, would be a great boon to this live attenuated approach, because it would suggest that such a virus can be safe in people and would provide some data to indicate that perhaps we should go forward with small-scale safety trials.
Some of this work has already been done.
Hi, David.
Hi.
Okay, how are you?
Sal is one of several long-term non-progressors being studied by David Ho.
Okay.
Ho has collected virus from a number of them and found they do in fact have attenuated strains of HIV.
We
we obviously have thought about why some of these individuals do so well. A few of these individuals have been looked at carefully, have critical mutations or truncations in certain genes, such as the NEF gene or other critical regulatory genes. And because of these mutation deletions, they are attenuated. We certainly think along that line that individuals who are infected for a long time with this type of viruses may actually be protected from subsequent exposures to HIV.
A live attenuated virus has protected monkeys, and by chance, a similar thing may have happened with some of the long-term non-progressors. The fact that they are alive and well more than a decade after infection provides some safety data. De Rosier thinks the live attenuated vaccine has enormous potential if the scientific community decides to test it on people.
I would like to think that this live attenuated approach is an approach that could save the world for AIDS. It's a vaccine that's cheap, would be easy to make, could envision it being distributed worldwide, and I could envision it being highly effective. I think that is something that could save the world from AIDS.
But NIH's chief AIDS scientist, Tony Fauci, thinks such talk is premature.
How's it going? It's going great. I feel good.
There's a ways to go before the biomedical research community and certainly the general public would feel comfortable with using a live attenuated vaccine. It may be that all the others will in fact fail, and we will have to take that chance with a live attenuated vaccine. I don't know that right now, but that certainly is conceivable.
But some medical ethicists question how long the developing world can wait for a totally safe vaccine. In parts of Africa and Thailand, some people face virtually a 100% risk of acquiring HIV. Women who depend on prostitution for a living might be willing to take a live attenuated vaccine if there was a chance it would protect them from AIDS. But is it ethical to try out a vaccine in the developing world that NIH scientists might not try out on themselves?
We have to take AIDS seriously for what it is. It is an enormously devastating pandemic disease. It will destroy a third, a half, or more of the populations of some country. We're talking about the ethics of human experimentation in the face of plague, not the ethics of human experimentation to try and restore hair on bald heads. If I am working in the sex industry in Thailand or Bangkok, if I'm exposed every day, whether I like it or not, to the AIDS virus, if I know that 100% of the people that I interact with are infected after a year, obviously my government has an obligation to try and stop those circumstances that lead to my exposure. But at the same time, some of these risks seem to be endemic to the culture. They're things that you're not going to do away with overnight. You might go to a subpopulation of mature people, competent people within there and say, look, here's the thing. We're not sure that we can wipe out the sex industry. We don't think you should be in it. But if you're going to stay there, we've got this vaccine that carries real risk, significant risk. But it can do you some good, we think. Here's the basis why. Do you want to sign up for a trial of this vaccine?
Kaplan believes that offering such a vaccine to people at high risk in the developing world is not racist, provided volunteers understand both the benefits and the risks.
What the researcher would have to do again is make that zealous effort to change conditions, make the real argument about why it's worthwhile given the lifestyle that's being led for them to think about this, assess the competency of the individual, and then say, look, it may involve experimentation on poor people in the third world because that's the nature of the risk they face. But we tried very hard and we're doing all we can to get them not to have to face those risks. If they continue to endure, it's not racist, it's common sense to try and do what you can to help them.
Closer to home, some in the gay community have strong opinions about a live attenuated vaccine.
It's crazy to think that we're going to solve this problem without taking risks along the way, and frankly, that we'll ever be able to solve it without losing some lives in the battle. Just like any other war, you know, some lives will be volunteered and lost. I hate to say that, but will be volunteered and lost along the way to the path of discovery. It's already happening in this disease. It's happened with every drug that we've developed. And it's going to have to happen with vaccines. I think Dr. Fauci and the others need to know that there is a large class of people out there, uninfected people, who are willing to take those risks, myself included, to make progress. You know, and I think we have to recognize on this ethical question that there's also an ethical question of not taking advantage of opportunities. You know, what's the ethics of letting 1 million more people get infected when we might have had a way to prevent that? And if in doing that we make a mistake and somebody gets hurt, that's what informed consent is all about. You know, there's a history for this, and I think we need some rational balance here and to stop letting, you know, excessive ethical concerns keep us from making progress in the disease. There's no ethics in that. Very ideal group right now are young homosexual men who are picking up this disease at an alarming rate again. But there are plenty of opportunities where there are high-risk people, high-risk behaviors that you're not going to fix by prevention messages and education. And I think we just have to get down to this task and stop fidgeting about it.
And if society decides that the live attenuated vaccine is too dangerous to be tested on the living, Kaplan has some other ideas.
One strategy that has occurred to me is that we might pursue some of the vaccine testing upon the dead. There are people who wind up in hospital who have suffered brain death. They are quite dead. There's no disagreement that they're dead and that life support and so on would be stopped for them. It may be due to accident or injury or gunshot or just terminal illness, but who might be willing, if you got their permission before they fell into such states, to be test subjects. So you'd have the option while alive of filling out a directive, and you could say, should I become comatose? I don't want aggressive treatment. And should I die with my brain ceasing to function? If you can keep my body going, I would be available for some fixed period of time to serve in medical research. I'd much rather be putting a cadaver at risk of transmission of a disease than I would a real live human being in Bangkok or Philadelphia.
But even if de Rosier's vaccine is tested in people, and even if it proves safe, it may not work. The HIV virus may find a way to escape science's best shot.
It is conceivable that we will never get a vaccine. I think we have to face that reality. People sometimes hesitate to talk about that because it gets taken out of context, and people say, well, they're giving up. No one's giving up. We're totally committed to finding lasting solutions to this terrible problem. But you must realize that it is conceivable that we will not be able to develop a vaccine. I personally think that we will, but I have to keep an open enough mind to say that it is quite possible that the nature of this virus is such that we won't.
Whatever the outcome of a live attenuated vaccine, the AIDS warriors are committed to continue their fight against the most formidable microbe in history. A fight that might not be won in their lifetimes.
The commitment to this obviously has to be sustained. Failures are not going to help maintain the enthusiasm of industry or government or what have you. But I don't see any other way of approaching it. I don't think we've ever had a target virus that looks anything like this, and we can't anticipate that it's going to be easy. And so I favor the notion that in time we will have a vaccine.
But the task of stopping the HIV virus is not the responsibility of scientists alone. Even without a vaccine, society had the means to prevent the spread of AIDS back in 1985.
What did people know as early as 1985? They knew how the virus was transmitted, by sex and by blood. They knew it was caused by a virus, and there was a test to identify those people who carried the virus. And also there was a prescription for how to avoid infection. Now, that is a tremendous success of science. What science has now been asked to do is give society an easy solution. Give me a cancer jab or give me an anti-AIDS jab. We can't do that yet. But that doesn't mean that in terms of finding a theoretically workable solution to this problem, we didn't do that. Science has solved that problem. Unfortunately, humans being what they are, society worldwide, I'm not talking about Americans, I'm talking about every society. Human behavior being what it is, it's a very difficult prescription to follow. But we do know how to do it. If you don't have sex with an AIDS-infected person, and if you don't share their blood, you won't get AIDS.
And I know a lot of y'all may think, okay, you're gay, that's why you got this. But it's no longer, you know, one time maybe it was...
Young people growing up in the age of AIDS cannot count on scientists finding a cure in their lifetimes. In programs like this one in San Francisco, HIV-positive volunteers tell students that the best way to beat AIDS is to avoid getting it in the first place.
And I tell you what, y'all have choices if you...
I think we've gotten more educated about it. We're a little more aware that it very much can affect us, that we are not immune to this disease. It's not going to pass us by because we're young. So I think we're definitely going to start protecting ourselves a lot more, which is very important.
I was 19 then, and I thought, this is an old man's disease. It's not mine. Besides, this is what I tell myself, I could tell if somebody had this illness. You know, they'd be sick. And I'm only going out with healthy people. So it's not going to happen to me, and it's not my problem.
Having sex without a condom is just as sure as using a guillotine, because eventually you probably run into that one person that has AIDS. And if you have a tendency to go out and have sex with lots of people, eventually you are going to hit that person, because AIDS is not as uncommon as people think.
I wonder why it's so important that we have to have a cure when it is preventable, you know? I mean, of course it's important, but, you know, it is preventable, and we have to remember that as well. And not just sort of rely on, oh, someday there'll be a cure.
While young people can fight the HIV virus with their own common sense, scientists must continue searching for HIV's weak spot, waiting for a lucky break.
I'm not saying that it's an impossible task. I'm just saying it's unpredictable outcome. There have been many serious problems humanity has faced before where we've had unexpected breakthroughs. That's what we will need at this point, because a straightforward approach won't work with AIDS.
For more information about AIDS, call the National AIDS Hotline. By special arrangement, their operators are now standing by to answer questions or to refer you to resources in your area. Call toll-free 1-800-342-AIDS. AIDS Research, the story so far, is made possible by the Aaron Diamond Foundation, supporting projects in New York City in minority education, culture, and medicine, including AIDS research, education, prevention, and the civil rights of people with AIDS.
Stay tuned now as Ruby Dee hosts another special broadcast, After Goodbye, an AIDS Story. That's next. Followed at 11 o'clock tonight by Independent Images from TV 12. Stay with us right here on public television for the Delaware Valley.